All right, hi guys. So this is the review session for exam 2, and I'm going to be covering lectures 14 through 16. Chantal will be covering the first half, which is lectures 11 through 13. So check out her video if you want to learn more about those topics. Okay, so to start off, this is just a little bit left over from what Chantal's covering, but to calculate the delta G of transport, which is like the free energy that we expect it'll either take or release to move different molecules across the membrane, we have this basic equation where delta G is equal to RT times the ln, the concentration of the inside over the concentration of the outside. And this is the equation specifically for transport into the cell. If we wanted to look at transport out of the cell, then we would have the same thing, except it would be concentration of A outside divided by concentration of A inside. If that's kind of confusing, or if you want a way to remember this, I like to think that, so usually we think of this as like the ln of the products over the reactants. So wherever that molecule is ending up will be on the top. So in this case, it's like on the inside, divided by where it started from, so the outside. And the equation is essentially the same if we have charged molecules, except now there's also this other term. Za is the like how charged it is. So for example, if we're talking about calcium, then that would be 2. If we're talking about chloride, that would be -1. And so these are like the charge on that specific atom. F is Faraday's constant, so that's just a constant. And then delta psi is like the voltage potential across the membrane. And so basically what this term means is now, let's say that we had a lot of sodium on the outside of the cell and not a lot of sodium on the inside of the cell. And we're curious about whether sodium will move across. But let's also say that we just have a ton of negatively charged ions on the outside of the cell. So in this case, even though moving sodium from the inside to the outside would be up its concentration gradient, since there's so many negative ions on the outside, the positively charged sodium is also going to be attracted to those ions. So this kind of takes that into account. And if the potential across the membrane is large enough, then we could actually see transport of things up their concentration gradient if they're charged. So just to do a quick example, first we're just going to read the problem and kind of think about what we would expect to happen, and that'll kind of at least make us check our signs, because I feel like with these types of delta G calculations, sign can be an issue. So we have 140 millimolars of sodium outside and 12 millimolars inside. So right off the bat, we're going to think, okay, if we're calculating the delta G for the sodium to move inside, we already kind of expect this to be positive since there's more outside and less inside. And then we also say the membrane potential is -70. So that also means that the inside of the cell is negatively charged compared to the outside. So that's going to make this even more favorable. So we expect that this is going to be a negative delta G because both of these things are kind of suggesting that the sodium is going to want to move in. So if we use our equation, we have to use the equation for the charge because sodium's an ion. And then we plug in all the values. We get -13 kilojoules per mole. And that's, like I said, kind of what we expected to get. So that's great. So what happens if instead of being a negative number, meaning it's spontaneous, what happens if it's non-spontaneous, but we still want to move these ions? In that case, we need to use a pump. So a pump is something that's going to use energy in the form of ATP to pump things against their concentration gradient or like force non-spontaneous transport to happen. And the basic structure of a pump is that we'll usually have 10 transmembrane helices, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then also these 3 modular domains. So we have the phosphorylation domain, the nucleotide binding domain, and the actuator. So the nucleotide binding domain is going to bind ATP. It's going to phosphorylate the phosphorylation domain, which will cause a change in shape. The actuator will also undergo a conformational change, and that will help allow us to move ions across their gradient. So that's a brief idea of what's happening. These are all the specific steps. So we're kind of starting here. This is like our enzyme. There's no calcium yet. So calcium comes in inside, and then ATP binds. And the nucleotide binding domain is going to have a slight conformation change you can see between these two. And then it will phosphorylate the P domain, and there'll be so phosphorylate the P domain. That'll cause a conformational change. We see calciums being released on the other side of the membrane. And then we also kind of see that the A domain is moving from here to here. So those steps are kind of happening at the same time. And that's just because like all these conformation changes are kind of going to happen all at once, but we split them up into steps just to make it like really easy to understand what all these different domains are doing. And then from here, once we have that conformation change, then the P domain will be dephosphorylated, which will cause another conformation change, and the A domain will reset. And then finally, all of these domains, so we can see that the N domain is still not in its original conformation. So finally, the P and N domains will also reset, and that will lead to this final state where it's ready to accept another calcium. So we can kind of see how this enzyme is like able to transport calcium and then also be restored to its original state and ready to transport more. There's other ion pumps besides this calcium pump, and they tend to have similar types of shapes. The other pump that was mentioned in class is the sodium-potassium pump. And this is like in all of our cells. The basic mechanism is that it'll pump out 3 sodium. So sodium is getting pumped out, and it will pump in 2 potassium. And that helps make these membrane potentials where sodium's high on the outside and potassium is low on the outside, and then inside sodium's low and potassium's high. And it's not just from this pump that these types of ions are, like the concentrations are maintained. There's also like channels and things, but this is like a really important pump that we have on all of our cells, and it helps maintain like that negative membrane potential that actually all of our cells have. Okay, so kind of moving on from the transport stuff, the second big topic we want to cover is small G proteins. So these are proteins that kind of act as like a molecular switch. So we see here's RAS, which is an example of a small G protein, and we can see that it is different in its GTP versus its GDP bound state. And these proteins also have an enzymatic activity where they can hydrolyze GTP. So that's basically how these proteins work. There's 2 transitions that they have to do. We have to be able to go from a GTP to a GDP, and then also from a GDP to a GTP. And the path we take for each of those transitions is different, and it's facilitated by 2 different types of proteins. So if we are in the GDP state, which is, in this case, they're calling it the inactive state, then we use a GEF, which is a guanosine exchange factor, which will take a GTP from solution and swap it with the GDP that's currently bound. So that's kind of shown in here where we see that like these, the GTP is being exchanged, which is like hence the name GEF. So now it's in its GTP bound form, but when we transfer back from GTP to GDP, we're actually just going to hydrolyze the GTP that we already have. So we see that we're still holding on to the same like molecule, we're just cleaving off that last phosphate. And that can help be facilitated by GAP, which is a GTPase activating protein. So like I said, these small G proteins are enzymes, so they are capable of doing this hydrolysis step, but they're not very good at it. So the GAP is just going to help stabilize that transition state even more and make it a more favorable reaction. So because G proteins are enzymes, they have a lot of the same things we talked about before the first exam that enzymes have. So they have a specificity region, which is like this base recognition region. So this will mean that the small G protein will bind to GTP, but it won't bind to like ATP or other types of nucleotides. It also has this P loop, which is a part of the protein that helps to bind the phosphates. And the P loop, which is found in other types of protein too, has this basic, basic amino acid sequence. So glycine, then anything, a glycine or a proline, anything, glycine, and a lysine. So the glycines make it really floppy, so you can see it's kind of able to just drape itself over this phosphate tail. And then the lysine is actually going to help stabilize the negative charge that's on the phosphates. You should know this general form probably, and what a P-loop is. We can also see there's a magnesium ion associated that helps to stabilize those phosphates in addition. And like mechanistically, the way that this protein is going to allow the cleavage of this last phosphate is it will take a water, activate it so it is able to nucleophilically attack this phosphate here, which is shown here. So like as you should know from organic chemistry, when that happens, this bond is going to go up and we'll get to this pentavalent metaphosphate bipyramidal transition state. And then after that, one of these will collapse and it'll break this bond. If that's like really unclear, just post about it in Piazza and I will clarify that. Yeah, and so the protein's going to like help facilitate this attack by the water using like different types of enzyme mechanisms that I'll talk about in a second. So what GAPs are doing is they're helping to increase the transition state stabilization. So when we look at this pentavalent metaphosphate bipyramidal transition state, we notice that we have lots of like these negative charges around the phosphates, around the phosphate because of all these oxygens. So to help stabilize this more, a GAP is basically going to insert an arginine finger. You can see it here. So this is the protein without the GAP. And then with the GAP, we have like all this extra stabilization, of the phosphates. And so, yeah. So the arginine finger is able to stabilize these negative charges. And then also the C-terminus of the GAP is able to help activate the water, like we said. By activate the water, I mean that, so if you remember with the chymotrypsin, we said that, okay, the serine is like not the best nucleophile. And so what we did was we made it, we deprotonated like the OH on the serine side chain. And then we're like, okay, so now this like oxygen anion is going to be a much better nucleophile. So by activating the water, it's the same thing. We're basically have some type of like acid-base catalysis where we will remove one of the hydrogens. So that will make this like an OH anion, which will be much more active and able to nucleophilically attack that phosphate. Yeah, so the catalytic methods of the small G proteins, they actually are able to like be examples for all the different catalytic methods we talked about before the first exam. So metal ion catalysis, because of that magnesium ion that's there, acid-base catalysis, so it's actually a glutamate that's going to hydrogen bond to the water, activating it. There's some transition state stabilization from the GAP. And then also catalysis by approximation, since like if we imagine this magnesium, this phosphate loop, etc., is going to really hold this nucleotide like in position so that water is able to attack. So all of these different methods are seen in G proteins. So next, we're going to talk about kinases, which are actually very, very similar to G proteins. But we say, okay, instead of using a water here, like a water to attack, what if we use like a serine or a threonine or a protein that has a alcohol side chain? And then what if, what if instead of GTP, we had ATP? And if we do those two changes, that's what a kinase is. So if we, sorry to keep going back and forth, but it's going to have a lot of these same types of things. So it's also going to have a base recognition region, except now instead of recognizing like a guanine, it's going to recognize an adenosine. And it's also going to have a P-loop. And it's also going to have associated magnesium ions. And so if you remember in class, he kind of drew protein kinase A. It had like a lima bean shape. So we're going to have like the N domain, or like the N terminus side of the protein is going to recognize the nucleotide with those, like with what I just talked about. And the C domain is going to recognize the protein that we want to phosphorylate. So the reason we need to recognize the protein is we don't want to necessarily phosphorylate every serine or every threonine or every tyrosine in the protein. We just want to phosphorylate specific ones, and in particular, protein kinase A will phosphorylate after this amino acid sequence. So like RR, X is anything, serine or threonine, and then a hydrophobic. So the C domain is going to recognize where these particular sequences are, and then it will like position the serine or the threonine for nucleophilic. And right here, I drew the transition state. So we see this OH is already like, has already attacked. So this is the OH from the serine or the threonine. We see the pentavalent metaphosphate bipyramidal transition state again, and then we can kind of see the different residues of the enzyme that are helping stabilize this. In particular, we see there's 2 magnesium ions. We also see a lysine, so this is kind of similar to like what the arginine finger might do, or this lysine, I guess, since this lysine might have been from the P loop, but let's say this lysine is like kind of where the arginine finger would have come in for the last, for G proteins. So this should look very familiar, and like this type of transition state and like the types of interactions, especially these magnesium interactions, are the same for kinases and G proteins. So kind of putting it all together, we have the epinephrine pathway, which is a signal transduction pathway that uses G proteins and these kinases. So just one thing before I start, before I was talking about small G proteins, which are G proteins that require a gap because they're not very good at hydrolyzing. This G protein that's associated with this receptor is not a small G protein. So it's a lot larger. There's 3 different domains, and it's actually capable of doing the GTP hydrolysis without a gap. So it doesn't need an extra protein to make it better at hydrolyzing the GTP. It's able to do it by itself. So just to run through how this pathway works really quickly, epinephrine is going to bind to this 7-transmembrane receptor, and the 7-transmembrane receptor bound to epinephrine will act as a GEF for this G protein. So we go from the GDP to the GTP form here, and then once it's in the GTP form, the different subunits will dissociate. The beta and gamma will stay together, but the alpha will dissociate, and it will find an adenylyl cyclase protein, and it will activate it. So what adenylyl cyclase does is it takes ATP and turns it into cyclic AMP, and the cyclic AMP is called the second messenger, and that will go into the cytoplasm, and then it will bind to the regulatory regions of protein kinase A. So normally protein kinase A has the catalytic and the regulatory regions, and you can see they're bound together so that the catalytic region is inactivated. But once this cyclic AMP binds, the regulatory regions change shape. The active protein kinase A is released, and then it will go do its job, phosphorylating proteins, which lead to the effects of epinephrine. So just a couple of extra things to note. This is called like GS, which is like G stimulatory. So this G protein is stimulating adenylyl cyclase. We also have GI, which is essentially the exact same pathway, except instead this G protein will inhibit adenylyl cyclase. So there'll be a different protein, a different G protein. It will be associated with a different receptor, and the receptor will have a different ligand. So there are like epinephrine will like cause all these effects. And then the GI, like whatever the ligand is that binds to the GI receptor, will cause the opposite effects or prevent the effects of epinephrine. Like I said, C-AMP is called the second messenger. There's other examples of second messengers. If you're interested, you can look in the book about the GQ pathway that uses a second messenger that I believe is called PIP3. And then also this binding of the pKa, you see there's like, for each regulatory region, 2 cyclic AMPs bind to the regulatory region, and those are going to change it allosterically, which I mentioned, but also this binding is cooperative. So once one binds, the other one is much more likely to bind. Okay, so some key takeaways. So remember that the 7-transmembrane receptor acts as a GEF for the G protein that's associated. Like I said, GS alpha does not require a gap because it's not a small G protein. It's just like a normal G protein. So there's kind of two different things, small G proteins and like G proteins. And the only difference is small G proteins are not as good at hydrolyzing the GTP, so they require a gap. And the reason we're using a G protein here to begin with is because it kind of acts as a molecular timer. So we know that eventually this like alpha subunit is going to hydrolyze and become inactivated. So that will stop the production of cyclic AMP. Also, like we will start degrading epinephrine, and then if we can degrade cyclic AMP, then like we can kind of shut this whole system down. And then that's what I'm saying here, that cyclic AMP is degraded by phosphodiesterase. And if you are curious, then here are the two reactions. So this is the adenylyl cyclase reaction, how we go from ATP to cyclic AMP. And here's the phosphodiesterase reaction, where we're using a water to go from cyclic AMP to AMP. And both of these mechanisms are essentially the same and also very similar to the G protein and the kinase reaction. So we have like either a hydroxyl or a water that's attacking a phosphate, and then that's leading to like cleavage of a bond. So it like pops up, pushes down, and then cleaves off. And it's the exact same here, which also means that this is another example of a pentavalent metaphosphate bipyramidal transition state. So it's not like, don't feel like that is something you have to memorize. Just like there's a lot of places that that same thing can come up. So if you really understand like why that transition state is happening, then you can recognize it other places. And by saying you don't have to memorize it, I mean like don't memorize it for every single reaction, but understand the concepts of why that type of transition state is happening, and then you can like recognize it even in things we didn't specifically talk about in class. And then finally, these multiple steps in the signal transduction. So you may look at this and be like, this seems really inefficient. We want to go from epinephrine binding to effects, and we have like a bunch of steps in between. And the reason we do that is because this allows it to act as an amplifier. Specifically, amplification occurs at any step where like a single activated upstream protein or like thing can activate multiple downstream things. So for example, this 7-transmembrane receptor can activate multiple GS alphas because like once it's activated, it dissociates. So we can imagine another one can come in and bind and then be activated. And then once it binds to this adenylyl cyclase, the adenylyl cyclase will make multiple cyclic AMPs. So this is not a one-to-one ratio. It's not like one adenylyl cyclase makes one cyclic AMP. So there'll be a lot of these. And then same for like protein kinase A. Once it's activated, it can phosphorylate multiple proteins. It's not only going to phosphorylate one protein. So at each of these steps where kind of multiple downstream things can happen, we can imagine that we can like amplify multiplicatively the response. So maybe like just a couple epinephrine bindings can lead to an like huge change in cellular signaling and like what the cell is doing. Cool. So I'm going to move on to like the final big topic for this section, which is nucleic acids. So we mentioned some nomenclature in class. I don't think this is like the most important thing to know. Nucleotides is the base, the sugar. I didn't write the sugar, but it's the base, the sugar, and the phosphates. And nucleosides are just the base and the sugar. And there's different names for the different, like each of the bases has different names depending on which form they're in. But what is important is that you should know all these structures, and also you should be familiar with this numbering system. So I don't think Professor Yaffe said this in class. Like he really stressed that you should memorize all the amino acid structures, but at least when I took the class, he assumed that we already memorized the nucleic acid structures. So he does expect you to know these, even though he didn't specifically say it. So make sure you spend some time to memorize it or put it on your cheat sheet. And then the way these nucleic acids are bound together, which you probably remember from like 701, is through these phosphodiester bonds. So, and we're bonding from the 3' hydroxyl to the phosphate that's associated with the 5' hydroxyl. And so, since this is called the 5' end because like yeah, there's like the 5' part. This should probably be bonded to a hydroxyl, not just the OH, but the 5' part is like the part that we're starting with, and then this is the 3' end because like we have the like 3' hydroxyl side. Yeah, so you should, I think this is also something you should have really learned in 7013, but like kind of be able to like recognize this. Maybe he might not ask this on this test, but it's like pretty important to be able to recognize what the 5' and the 3' ends are. And then, as you know, the reason that DNA kind of works the way it does is because it has like this type of base pairing. I'm just gonna move this. This is called Watson-Crick base pairing, just because like now we've started learning about like Hoogsteen pairs and like wobble. So we know that there's other types of base pairing that these can do besides just this, but this is like the very traditional like base pairing. So we know T goes to A, C goes to G. You should know like how to draw this and this. So like either memorize it or put it on your cheat sheet. And like the really important thing about like the fact about these base pairs is that there's the same C1 to C1 distance. And by I think this should be C1 prime and C1 prime. Yeah, and what I mean by that is like that the carbon that's associated with the sugar is going to be the same distance apart. And so that means we can basically have like a sequence independent geometry. So like DNA is not fatter like in regions where there's AT bonding versus GC bonding. It is able to be like the same structure regardless of what the actual bases are. And then the other thing that is really important that you know is about these major and the minor grooves. So the major groove is sequence dependent. So what I mean by that is like, say we have a T bonded to an A, then if we read off this series of like hydrogen bond acceptors and donors, we have methyl, methyl, acceptor, donor, acceptor. If we have AT, it's going to be acceptor, donor, acceptor, methyl. So those are like, it's different depending on which way you read it. And same for CG is also different. So it's hydrogen, donor, acceptor, acceptor, and then it's different the other way. So if we gave you a pattern of this, you should be able to say, is it TA or is it AT or is it GC or CG? So it's different between TA and CG and also different between TA and AT. So that's what we mean by like sequence dependent. And so proteins like transcription factors who care about what the sequence is and only bind to certain sequences can read the major groove and say, oh, I know like what this sequence is. The minor groove, so is different. So this is, so this is going to be acceptor, donor, acceptor. And this is acceptor, hydrogen, acceptor. So even though there's like this difference between like hydrogen and donor at the second position, like in like for all intents and purposes, like these are basically the same sequence. And so proteins will also probe this. So say we have a protein that binds to DNA, but it doesn't care what sequence of DNA it binds to. It can probe the minor groove and know that it can probe the minor groove and know that it's bound to DNA or to like nucleic acid string. So yeah, so proteins will probe both the major groove and the minor groove, but they will probe them for different purposes because of the sequence dependence and sequence independence. Okay, so in this one, in this part, I'm talking about probing like DNA. Here, I'm talking about recognizing single bases. So when we want to bind ATP or GTP, this is not like a nucleic acid like polymer. This is just like the single nucleotides themselves. And so ATP binding proteins recognize N1 and N6, and GTP binding recognize N1, N2, and the O6. So yeah, this is why like it's good to know these things. So yeah, I probably should have like drawn this out, but as an exercise, you can draw out and like see exactly like what these N1, N2, O6 correspond to. And then you can realize that these are actually like the same areas that are involved in base pairing, which is like interesting. So basically the things that allow like G to bond with a C and an A to bond with a T and not the other way around are the same things that allow proteins to recognize and distinguish between an A and a G. And then also GTP binding proteins, in particular have this sequence NKXD, where like the D is recognizing N1 and N2, and then the N is recognizing the O6. And like same with the P loop, everything X always just means it can be any amino acid, it's not conserved. Okay, and then, sorry, we're getting towards the end, so thanks for hanging in there. So the DNA double helix, these are some features that you need to know. So obviously it's a double helix. You should know that it is a right-handed twist. Yeah, which basically means if you curl your fingers in the direction that it's twisting, your thumb will point in the direction that the strand is going. If you use your right hand. If you use your left hand, it won't. So that's what we mean by right-handed twist. That they have anti-parallel chains. And then here's some of the dimensions. So it takes 34 angstroms. If you remember Professor Yaffe's like spiral staircase, where you're at the exact same position horizontally, but you are like one level up, and 20 angstroms in diameter, 3.4 angstrom rise per base pair, which means that there are 10 base pairs per turn. The bases are inside the helix and they're perpendicular to the backbone, and the phosphate backbone's on the outside, which makes sense because the phosphate backbone is very hydrophilic, so we'd want that to be exposed to the outside, and also the phosphates can help protect the DNA from nucleophilic attack, which I just think is really cool. And you always have a purine bonded to a pyrimidine, and the reason for that is specifically because of this same like C1 prime to C1 prime distance. If you had like two purines bonded together, the DNA would be really fat in that region, and then it wouldn't have all this nice structure, so that's why we only see purines and pyrimidines bonded together. And then also to mention, the DNA helix is very stable. So it's mostly stabilizing by these overlapping pi clouds. This image is from benzene rings, but you know the pi orbitals are like above and below the plane of like the ring. So you can see how like you can have these pi clouds that are overlapping, and then this can lead to favorable interactions. Yeah, so that's like the main stabilizing force. And like that kind of makes sense. You see how these like A, T, like these bases are really stacked on top of each other, so there's going to be a lot of overlap. And so this makes it very favorable and makes the DNA very stable. Also, van der Waals forces play an effect as well. And like this is really strong. So if there's even just 100 bases, you have to boil the DNA to separate it. So that is a lot of energy it takes to pull those two strands apart. We're not even breaking any covalent bonds, but just to kind of like pull those apart takes a lot of energy, which is like, yeah. So because of this, strand separation is called melting, and then going from two single strands to a double strand is called annealing. If you remember when we talked about like tryptophan and tyrosine and how they absorbed UV light because of their pi electrons. So the same is true for DNA. So we have absorption, UV absorption from DNA. That's how like things like a nanodrop work, if you've used those in lab. And there's a different absorption from single and double stranded. And that's because of these overlapping pi clouds. You will get like, you get less absorption for the double stranded DNA because it's more difficult for it to go from that pi to pi star transition because of these interactions. You don't need to know that. I just think it's really cool. And so because of this, you can actually measure like this change in absorption over temperature, and you can get these really nice melting curves. Yeah, and then also like the more strongly that the DNA is like held together, which is like increased GC content, you'll see a shift in the melting temperature, so it will melt at higher temperatures. You can also probably imagine that if like you get longer and longer sequences, they will also melt at higher and higher temperatures as well. Yeah, So that's all I have for the review session. I hope it was helpful. Feel free to email me, or it's probably better if you put things on Piazza so everything can see them. If you have any questions or if anything was confusing. But good luck on the test, and I hope you guys are all doing well.